Okay, so the first thing, we looked at mitosis and meiosis. If we just bring up mitosis, what was the goal of that, Maddie? And make two identical cells. So if we have our normal cell, mitosis just creates two identical cells. So if this is A, then we're going to have exactly the same. Two identical cells. So we need to go through a process of duplicating our DNA and then ready to divide that into two new cells. So mitosis basically stands for dividing the nucleus, dividing the material of the nucleus. Okay? So we get two identical cells. Meiosis is slightly different. Its goal isn't to create two identical cells. Oh, and what's meiosis' job? It creates 4 cells, but that's not its primary goal. What would happen if one of these... Well, first of all, how many chromosomes are in a normal cell, Owen? 46. Good. 46 chromosomes. Okay? So if we have 2 cells, Owen, that are both 46, and let's say one's a sperm and one's an egg, and we combine them, how many are we going to get? 92. Is that going to be a problem? Yes. I mean, we can see if we just have one extra chromosome, chromosome 21, we're going to get Down syndrome. So it's very important that we get exactly the correct number of chromosomes in a new individual so that when they develop, they've got all the chromosome material they need and they don't have any extra. So meiosis' job is not to create 4 cells, but it's to create cells that, when they join together, are going to be able to fuse and create our full complement of 46. Yes?
Can chromosome 1 less leave?
Just as dangerous. You don't generally see that as much, but there probably would be cases of that happening. I'm just not sure whether they would reach full term or not.
What would you get?
Most of the time, you have a chromosome and it provides a level of, say, protein or something like that. So just having one less, sometimes you might be able to get away with it. I'm not sure. We could look into that. But I know there's lots of syndromes that have one extra, for example, Klinefelter or Down syndrome, and that has devastating effects. So I would presume that one less would have the same. Okay? So what we basically want to do is go from one cell to basically 4 cells, and each of these 4 cells have only 23 chromosomes. 23. Okay, so that's meiosis. And they could be either sperm or an egg cell. We call these cells with the full complement, full 46 chromosomes, one set from mum, one set from dad. We call them diploid. And we call the cells that only have half the number of chromosomes, so not a full set from mum, not a full set from dad. We call them haploid. And the way I remember it is haploid I think of as half, and diploid, di, means 2. So 2 sets. Okay? So if we look at this in a bit more detail, would you like more time to get that? Yeah. There it is. I'll pause. Okay, so the one that we're going to focus on today is meiosis. Okay? So basically, meiosis starts very similar to mitosis. The cell is in a bit of a cycle. We call this the cell cycle. So it's... Oh. So we call this a cell cycle. We draw it as a circle. So it goes around this circle and it's divided into sections. So the first section here, in this case, we call G1, S, G2, and then this section here could either be mitosis or meiosis. Okay. So G1 is where we have just the general growth of the cell. S is really the only important phase for us. That's DNA synthesis. So that's where our DNA doubles. Okay. So in a cell, hopefully I can rub this stuff out over here. So basically, in a cell, we have the nucleus, and it looks like this. All the DNA is just all spread out, and it's everywhere. So in the synthesis phase, all that happens is we make a copy of that DNA material. So we make a copy of it. Okay, so we're making a copy of all the 46 chromosomes. Okay. And then we're ready to go into mitosis or meiosis. Okay, so once we've replicated the DNA in the DNA synthesis phase, we've now got 2 copies of our DNA and we're ready to go on to divide. Now this will happen in mitosis or meiosis. But because we're focusing on meiosis, we'll go on to that one now. Oops. So meiosis. Okay, so here's our cell. Here's our nucleus. Now, this phase here is called Prophase 1. So meiosis, Prophase 1. So we have our 23 copies from mum. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. And we've got another copy from Dad. So there they are. So they look like this. Now, because they've been replicated, they look slightly different. Okay, because they've been replicated, they've now got a second arm to them. Okay, so each one of these chromosomes have a second arm. Okay, we call those chromatids. Okay, so each one's got a second arm, and the same with the blue ones. Whoops, with the blue as well. They're going to have a second arm as well. Where they attach is called the centromere, like so. Okay, so in prophase 1, we start to see that happening. With the chromosomes condense, they're being replicated. Okay, so they look like they're Ks, or the Xs, and they're condensed. They're paired up with their partner. So chromosome 1 from Dad and chromosome 1 from Mum pair up together. Chromosome 2 from Dad, chromosome 2 from Mum pair up together, and they join really close to one another. Okay? Why they do that is a bit beyond what we need to know, but it basically allows them to sort of swap material between themselves. But for us, it's a bit beyond us. While that's happening, the nucleus is dissolving. Okay, so imagine that nucleus is starting to disappear. I wish I could do it with my eraser, but I can't. So that starts to disappear, and we get our centrosomes moving to opposite poles. Okay. The next step. Okay. So the next step is we have now got no nucleus, and our chromosomes start lining up together. So we've still got our centrosomes here, and they've got their microtubules or spindle fibers. They've joined up to those chromosomes, and they've lined them up across our metaphase plate. The only difference is the chromosome, let's say chromosome 1, chromosome 2, chromosome 3, chromosome 4 and chromosome 5. I'm just going to pick 5 to make it easier to see. And then the ones from Dad line up this way. So this is different to mitosis. Hopefully you can see that. In mitosis, they would all line up in a line. It would be like this. There's no distinction. There's no grouping up side by side. They would all just line up in a straight row. This is key to meiosis. Okay, and you'll see why in a sec, hopefully. So this is called metaphase. Metaphase 1. And it does this so that as we drag these across, each side only gets either chromosome 1 from mum or chromosome 1 from dad. They don't get both. Can everyone see that? It's either dad's chromosome 1 or mum's chromosome 1. It's either dad's chromosome 2 or mum's chromosome 1. So the side that these line up on is random. Okay, it's independent. Okay, I guess I shouldn't really call it random because it's a very controlled process, but it's independent. So it means that they could be on either side. So that means we could have different combinations. And that's why we don't look like our brothers or sisters. Yes, Jan?
So is that 5 or 10 different chromosomes?
That is 10 different chromosomes, but they're all... So these are chromosome 1 pair. They're going to be slightly different. But most of the genes of them are going to be the same. Well, the genes are going to be the same, but they're just going to be slightly different. So this is chromosome 1, chromosome 2 pair, chromosome 3 pair, chromosome 4 pair, and chromosome 5 pair. Okay, so they're going to be slightly different. This is how we get the relationships between genetics. Do you have attached earlobes or unattached? Do you have dark skin or light skin? This is because of the chromosomes that you've got from your mum and from your dad. Okay? So then we move into anaphase. Okay, so anaphase is now the centrosomes have pulled them apart. So you imagine these spindle fibers are actually attached to all of the chromosomes. Okay. Now these centrosomes are pulling them apart, back to the poles. Okay, so if we do that, we're now going to have red, 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 red. And on this side, we're just going to have the one red. And then we're going to have blue, 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 and one blue. Okay? So they've been pulled to the opposite sides of the cell. So, and that's called anaphase. Okay, so now we start to pinch in. Okay, we start to pinch in, and we start to form a nucleus around these new chromosome sets. So remember, this one's going to have blue, 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 blue. And this one's going to have one red, 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 red. Okay. So you don't need to get too caught up on the colours now. The colours are only to show you that homologous chromosomes either go one way or the other. Okay. And that they line up together. So this process is called telophase. I'll do it back in black. Telophase 1. We then get cytokinesis. And we've got now 2 cells. Okay. Now I'm just going to follow one of these down through another division. And it's going to be this one here. Okay. So this one's got... Okay. So now we've got 2 cells. So now we can move one of these down. So we'll pick this one. Okay, so now what happens is the membrane dissolves again. The nuclear membrane dissolves again. Centrosomes are still ready to pull them apart, just like mitosis now. Very similar to mitosis. Basically exactly the same as mitosis now, except instead of separating 46 chromosomes, it's now going to separate the only 23 that are there. So this is prophase 2, and we've got our 4 red and 1 blue. Okay, so now we get the centrosomes moving them into the middle. Okay, and this is called Metaphase 2. And then they get pulled apart. So you're going to have 1 red, 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 and blue, red, 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 blue, and we're going to have our centrosomes attached and pulling them apart. And remember, they're attached at the centromere. Okay, and this is Anaphase 2. And then finally, we get our Telophase 2 and cytokinesis, and nucleus starts reforming. The chromosomes will start to expand, but on this side we've got red, 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 and blue, and on this side we've got red, 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 and blue, and then eventually we've got 2 cells. Okay, so if we zoom out on that, okay, this is going to happen exactly the same on this side as well. going to happen exactly the same here, and then it's going to give us 2 more. So just to recap, okay, the goal of meiosis is to replicate the DNA of the nucleus and then divide it into haploid cells, dividing that nuclear chromosomes in half. So we start with prophase, nucleus dissolves, centrosomes move apart, the chromosomes condense, they've already been replicated, homologous chromosomes line up, so the copy from dad and the copy from mum line up. In Anaphase 1, the homologous chromosomes get pulled apart to opposite cells. In Telophase 1, we get the cells forming back together. In Prophase 1, the chromosomes nuclear membrane dissolves again. Centrosomes then in Metaphase 2 move all chromosomes into the metaphase plate. The spindle fibers have attached to the centromere and pull our chromosomes apart. Telophase 2 and cytokinesis happens, creating 2 cells, and that process has occurred on the other side as well, creating another 2, so we've got 4 sperm cells. And that's happening all the time. Gentlemen, you're making hundreds of thousands to millions of sperm cells per second. Okay? Each one of these could be responsible for a baby.